Welcome to this virtual talk. I am Marijne, a PhD researcher in the lab of Professor Anne Goris at the KU Leuven in Belgium. This presentation is entitled Smoking and Multiple Sclerosis Risk, a Mendelian randomization study. Observational studies suggest that smoking is a risk factor for multiple sclerosis, with smoking increasing the risk for MS with 50% in comparison to non-smoking. However, observational studies are subject to reverse causation and confounding. It may be that the onset of disease causes an increase in smoking rates, and this is an example of reverse causation. Another issue is confounding. Confounding occurs when both smoking and multiple sclerosis susceptibility associate with a common cause. Therefore, my research question is whether smoking is indeed a causal risk factor for multiple sclerosis. To investigate this, I make use of the Mendelian randomization framework to overcome the issues of reverse causation and confounding. Instead of asking people whether they smoke or not, genetic variants are used that are robustly associated with smoking. By estimating the effects of these genetic variants upon multiple sclerosis risk, we can derive a Mendelian randomization estimate for the effect of smoking on MS risk. Two-sample Mendelian randomization is used, in which the SNP estimates are derived from large genome-wide association studies in two independent populations for smoking and multiple sclerosis risk. Besides the three smoking phenotypes, smoking initiation, cigarettes per day, and the lifetime smoking index, I also investigated whether BMI is causally associated with multiple sclerosis risk. In our Mendelian randomization analysis, a genetically predicted increase in BMI was associated with an increased risk for MS with 31%. And this is in line with other published Mendelian randomization studies. In contrast with BMI, there was no evidence for causal effect of a genetically predicted increased likelihood for smoking on multiple sclerosis susceptibility. The Mendelian randomization effect estimates were consistent across all the different tests performed. Likewise, a genetically predicted increase in cigarettes per day or the lifetime smoking index did not associate with multiple sclerosis susceptibility. Finally, we performed a multivariable Mendelian randomization analysis, in which we estimated the effect of BMI on MS risk after conditioning on smoking initiation. The association between BMI and MS risk remained significant after conditioning on smoking initiation. In contrast, the effect of smoking initiation on MS risk remained non-significant after conditioning on BMI. In conclusion, our Mendelian randomization analysis for smoking phenotypes do not support a causal association with multiple sclerosis susceptibility, and this is in contrast with observational studies. Mendelian randomization is especially helpful for multiple sclerosis to investigate whether an environmental factor such as smoking is etiologic in the disease, as we don't know when the disease starts exactly. Furthermore, it overcomes confounding and reverse causation. But it also has its limitations. Weak instrument bias cannot be fully excluded, and it is limited to linear associations. Future challenges are to investigate whether interactions between environmental factors and multiple sclerosis susceptibility are important, whether their effects are time-dependent, and finally, we need to move beyond susceptibility and investigate the effect of these environmental factors on the disease after disease onset. I'm happy to discuss the research results in more detail with all of you. Thank you for listening.